Hello, everyone. Can you see me? Can you hear me? Someone give an indication that you all can hear me. Hello.
Yes, sir.
Can you hear me now?
Yep.
Loud enough?
Yes.
Okay. Thanks, Subbu. I will take care of from now on.
All done.
Hello, test 1, 2, 3. Huh? Loud enough. Yeah. Finish చేయలేదా? Ask them to finish them. And then tell me.
Yeah, if all done, submit your options. Not done.
rendering now. Oh, that same parts. Check clicking frames. Make sure the right directions.
All done. Very good. You can take the number.
Can I start?
I'm doing the one. There's like not very much.
Hello everyone. So today is a little bit unconventional. I'm teaching using Zoom while sitting in the next room. Okay. So when there is no crossover between the homologous chromosomes, it looks like this. The blue and green are two homologous chromosomes or sister chromosomes. Okay. There is no crossover here. So they just remain uncrossed. So all four chromatids that are resulting from meiosis will be there. Whereas crossover can happen. Here the crossover occurred. The blue chromosome and the green chromosome are touching each other. So this place is called chiasma, so at which the crossover will happen. As a result of crossover, we have recombinant chromatids. So the chromatids that have undergone chiasma or crossover, that only will be recombinant chromatids. But other chromatids that did not undergo the chiasma are non-recombinant. So these two are non-recombinant chromatids. So as a result, 50% of them are recombinant chromatids. So the ones that have undergone recombination, yes. Oh, I need to record, sorry. Thank you. Yeah. Let me check one more time carefully. Yes. So
Recording in progress.
okay. So this is okay. Do you see any results? Oh yeah. Okay. Okay. So once the recombination happens, we have the chromosomes that are mixed up with green and blue. Okay. So the map distance, we know how to measure them, that we need to understand the recombination frequency. How can we measure the recombination frequency? We need to have a test cross. Okay. When heterozygote is crossed with a homozygous recessive one, and then we can see in the segregation whether they are freely recombining or not. If they are independently assorted, then we have to see one-to-one segregation, one-to-one segregation, 4 ones. Whereas if they are on the same chromosome, then the recombination frequency will be normal in the parents, that is 1-to-1, but whereas the recombinance will be much less. So I will again show you more examples of that. And then the recombinant chromatids, especially if we take about 50 different meiosis, out of 50 different meiosis, one chromosome, one meiosis is the one that recorded crossover. Out of 50, if we assume just one had meiosis, one meiosis had crossover, that means only 2 chromatids out of the 4. Even the one that has undergone the crossover, the recombination happened only in these 2 chromatids, because here is the crossover, chiasma. Okay, so for each chromosome, each chromatid, there are 49, 49, 49, 49. All 49 meiosis of 49 independent non-recombinant chromatids, plus 2 more chromatids. So see, the total number of chromatids are 200 out of 50 meiosis, only 2 chromatids have undergone recombination. So 2 out of 200, that gives 1 centimorgan. And then last class we mentioned little bit about double recombination. That is, recombination is happening here. Had there not been the second recombination, we could have seen clear recombination for small b and capital B here. Capital B might have been here, small b might have been here, if there is one single crossover. But this double crossover happened. A crossover happened here, a crossover happened here. One chiasma occurred here, and another chiasma occurred here. Okay, so this one nullified the crossover. This looks like totally non-recombinant for A and B, though the middle segment is swapped. The middle segment from here to here, the green segment went up, blue segment came down, but with reference to crossover, it is clear. There is no problem here because the parental alleles, capital A, capital B, are together even in the recombinant chromatid, because only the middle part got swapped. If you have understood it, then I would like to show you one important thing. When we talk about 3 different genes, like LG, GL, SU are 3 different genes. Let me show you a whiteboard. How to show whiteboard? Where is that? I want to draw something. So the maize plant grows straight like this. If it is the corn plant, it is growing straight. The leaves are here. Oh, it's disappearing for some reason. Yeah. So this is the normal erect plant, erect corn plant. Okay, but some plants grow like this. This is a mutant. So this plant is called lazy plant. Okay, so this is called lazy. It's a mutation. Because of mutation in stem, the plant is kind of falls down like this. And then there is another plant called sugary. So the normal plant have the leaf, the corn leaf, the glassy leaf. The normal corn leaf is something like this, the leaf. And this leaf is full of spines. If you see it, it is full of trichomes, tiny spines. If you rub this, sometimes your skin get punctured. This is called spiny. But the mutant plant, that is not spiny at all. That is completely glassy. Glassy in the sense smooth surface. Smooth or glassy. Okay. So there is one mutation in maize that can cause stem as if the plant is falling down. So we call that mutation as lazy. And then another mutation is glassy. Another mutation is the stalk. Generally, this maize stem, if you chew it, it is blunt in taste. But this mutant plant is sugary. You chew it, it is sugary, very sweet. That's why it is sugary, because sugar gets accumulated in the stalk, which is not normal, a mutation. So there is this cross can be made, a plant that is capital lazy, say capital glassy, capital sugary. So similarly, the homozygous plant lazy, glassy, sugary. This is a dominant plant. It's a normal plant, normal looking maize plant. If this is crossed with a plant which is completely lazy, completely glassy in the sense recessive for all three traits, lazy, glassy, sugary. So what has happened here? A cross is made between a plant which is normal, that's why it is capital LG on both homologous chromosomes, a glassy, which is normal leaf, capital GL, is normal leaf with all spines on it, and then sugary, because it's not mutant yet, so the stalk is blunt. You chew it, nothing, the tasteless stalk. Only cattle can chew it. So this is crossed with a mutant. So this one is lazy, the plant falls down, glassy, slippery smooth, and then sugary. If you chew it, that is sugar. So there are three different recessive genes together. A cross is made. So what would be the F1? LG, glassy, sugary, LG, glassy, sugary. So this is the heterozygous condition. Okay. So one chromosome is coming from here. One chromosome containing all three recessive alleles is coming from here. Then it is heterozygous. So this heterozygous plant, we can cross with a totally recessive plant, which is like this. Is crossed with lazy, glassy, sugary, lazy, glassy, sugary. Okay. So let me go through again the whole thing. The normal corn plant that farmers grow in field is a erect plant. Stands up. But there is a mutant that is called lazy. So the dominant allele for lazy, which keeps the plant erect, is called capital LG. Okay. So if it is a mutant plant, we can call it a small LG. Similarly, the glassy, normally the corn plants are not glassy. They have spines. So if there is a glassy plant, that means that is a mutant. So only glassy, small glassy gives you this glassy plant. And then this is sugary. If you chew the stem, that is sweeter. that should be recessive, both small su, small su. So this cross is made. This is called test cross. So now I hope you can see this PowerPoint. So these are the test cross progenies. Okay, so 3 genes are segregating. One thing, whenever we see these 3 genes are segregating and somebody is showing this kind of data, we need to see whether all the different recombinants and the parental types, whether they are all same, equal. Like if there are about 700 plants, the parental types and recombinants should be exactly equal. They are not equal. These parental types are more and recombinants are very less. This indicates that these 3 genes are located on the same chromosome. We'll come back to this statement again, why I am saying that these 3 genes are located on the same chromosome. So in this test cross that I have shown, if the recombination might have happened, it could be LG, glassy, sugary, small sugary. So why did we get this? Because the recombination happened between the 2 chromosomes. That is, let me draw again the test cross. LG, glassy, sugary chromosome. When it is crossed with small LG, glassy, sugary, like the test cross I mentioned. So if the recombination might have happened LG, glassy, sugary, so this plant is called sugary. We can name it as sugary because for lazy, this is lazy. For lazy and glassy, it is normal, so it is sugary. And we call lazy, glassy, and sugary. This plant we call for erectness, it is a normal plant, but for glassy and sugary, it is mutant. So these plants we call glassy and sugary. Okay. And then if any plant is lazy, glassy, and sugary, this is generally is called lazy, glassy, and sugary, like the parent. Okay. So if any plant is just this one, and then this and this are there, this plant we call it as lazy, just lazy. For glassy and sugary, it is normal. And then if there is any plant lazy, glassy, and sugary is normal, then what do we call this? We call this plant lazy, glassy. It's not sugary because sugar is normal. So if there is any plant like this, and like this is simply glassy. Okay. So how did this sugary plant appear? Because recombination might have happened. Say this is the lazy sugary one, and then how do I change the color? Say this is the chromatid of small lazy glassy sugary one. So if the recombination happened here, for example, there, Then we will get a recombinant chromatid, which will look like this. Okay. So this one got swapped here. Yep, finally I got. So because of this recombination, the fragments changed. This red got this magenta color one, and the magenta color one got red fragment. Okay, so if there is LG, glassy, and sugary chromatid has undergone recombination with LG, glassy, and sugary. So the recombination is at this point, the sugary went up and the glassy came down. Okay, so because of such recombination, we have different things. Here is glassy, that means GL alone is in the center. Okay, so you have lazy, sugary, or normal. That means something middle one should have been swapped here. But here it is lazy, glassy is fine. The sugary is alone small allele or recessive allele, making the plant sugary. Here is the lazy, glassy or recessive alleles. Sugary is the normal allele. And then lazy, sugary are the recessive alleles. Glassy is normal allele. And glassy and sugary are the mutant alleles or recessive alleles. And lazy alone is the wild type or normal allele. Here is the parental type, lazy, glassy, sugary. All three loci are mutant type alleles. So what are we trying to do here is trying to understand what is the order of the genes. You may say that, well, you already drew the order. This is not the order. This probably just a hypothetical order. But looking at this segregation, these are the test cross segregants. We are trying to figure out what is in the center. whether lazy is in the center, or sugary is in the center, or glassy is in the center. Okay. So we need to see these numbers. These are 4 and 2, very least segregants. You see, rest of the segregants are 33, 59, 44, 40, quite a large numbers. Whereas these two are very small segregants. And what is their phenotype? Sugary. That means just sugar is changed, but lazy and glassy are same. Okay, so that is roughly this genotype, but sugar alone is changed. And whereas this one, lazy glassy. What is lazy glassy? Lazy glassy are totally two recessive parents, and then this sugar is changed. So why do we assume sugar is in the center? Here I am making one important point. You know, you remember first time when I told about double recombination. In double recombination, I told you guys that the middle segment is swapped because of this double recombination. So one recombination happened here, another recombination happened in very close proximity. This can happen very rarely, isn't it? Two different chiasma occurring next to each other is a very rare event. Okay, so one chiasma itself is a big story. Out of 50 meiosis, we have seen only one recombination earlier. So out of 50 meiosis, only one undergone recombination here. So having this kind of double recombination is very rare. And what is in the middle? If the middle segment of dominant allele is swapped with recessive allele, that can be seen as a middle allele. That can be noted as middle allele. What I'm trying to say here, sugary has just 4 segregants, and then lazy glassy are just 2 segregants. So one parent is lazy, glassy, sugary. If the sugary capital is here, this is not the story, because the middle one will have the one that has undergone double recombination. Because we know that the double recombinants are least in number. That can happen very rarely. These are all single recombinants. These two are one parent kind, this is another parent allele. These two are one parent kind, and this is another parent allele. These are two are recessive parent alleles, and this is dominant parent allele. And this is dominant parent allele, these two are recessive parent allele. So, because these are the parental segments, you see LG, glassy, sugary, LG, small LG, glassy, sugary are parental alleles. So, what is in the middle should be sugary. Reason is lazy, glassy is there. If it is sugary is at the end, we should have noted large number of segregants, not just 4. 4 and 2 are very least. This probably occurred because of double recombination. Because double recombination happens very rarely. See, comparatively to 33, 59, 44, 40, the recombinants are just 4 and 2. So what is the thing that is recombined? Is sugary, because rest of them are dominant alleles. So sugary should have been in the center. And similarly, lazy and glassy, this is coming from this parent. Let us see if it allows me to draw here. The order is, that's good. So the question is, we need to interpret about this and then about this. If the order is LG, sugary, and glassy, that is crossed with the recessive parent LG, sugary, and glassy. So the recombination might have happened between this sugary went up here. This sugary went up here. And then this sugary came down. That fits in this phenotype, sugary. This is sugary. For glassy and lazy, it is normal. And lazy and glassy, it is recessive, whereas SU is sugary is dominant, normal. So that tells us double recombination happened here, and sugary is in the center, and lazy and glassy are either side. But we do not know lazy is this side or this side, or glassy is this side or this side. That part we do not know. So we just can identify with 3-point analysis. When we take 3 genes at a time in the mapping, that is called 3-point analysis. So what did we do in 3-point analysis? Creating a heterozygote that is between capital LG, capital GL, capital SU with small LG, small GL, small SU. And then this heterozygote is crossed with a total recessive parent, that is LG glassy sugary, LG and glassy sugary. And then the recombination happens in all combinations. Okay. So the least recombinants are the result of double recombination. So the double recombination swaps the middle segment. So what is the middle gene? That is the sugary. What is the other one? Lazy glassy, which is flanking, that is coming from the other parent. That clearly tells us sugary is in the center and lazy glassy are at the end. If you don't understand it, please meet me. Again, I can discuss with you in my office. Yeah. So altogether different topic we are going to discuss, folks. So the first one we discussed about double recombination that happens very rarely. So looking at the double recombination and segregating segregants, we can identify which gene is in the center. That was the first part of this topic. And second topic is, how do we do mapping in humans? Most importantly, most of you are medical doctors or medical professional, future medical professionals. So you need to know how the genes are mapped in humans. You cannot make crosses. You cannot make huge number of progenies in humans or the animals for that matter. Even if you mate 2 different cattle, female and male cattle, cow and ox, cow and bull, probably you don't get many populations. For each gestation, you will get only 1 child, right? Pigs can produce a little bit more. Maybe 16 or 17 pigs, or dogs can produce more. So humans are one or twins only. All the mammals will produce twins. So we don't have sufficient mapping populations to understand the genes, the order of the genes. So the DNA sequencing is used in this case. Once we have the sequence data, so sequence data of X and sequence data is Y, once we obtain that sequence data, we can map it to the reference genome, reference human genome. So in the gene banks, we have that reference human genome, and we can infer polymorphisms, genetic polymorphisms. Like what X has a nucleotide at a given position and how the Y differs for that position. So these are called polymorphisms. So these polymorphisms are quite prevalent on average anytime. If you take two chromosomes of unrelated individuals, you can see at least one difference in 1,000 base pairs. That is quite a bit. So you and me are unrelated. For any chromosome, we can see at least one difference for every 1,000 base pairs, one nucleotide or one base pair for every 1,000 nucleotides. That's quite a bit. So some of them are single nucleotide substitutions. You may have A at one place. I might have G at another place, same place, right? You may have C at one place. I might have T at another place. So that is SNP in short, single nucleotide polymorphism. Or we may differ copy number variation. We may have number of genes difference that you may have, say for a given gene, you have 20 genes. I may have only 30 genes. That is copy number variation. I will discuss how profoundly that impacts phenotype shortly. Or just repeats, that is dinucleotide repeats. Dinucleotide in the sense two nucleotides at a time, say adenine and guanine, or cytosine, thymine, or thymine and adenine, or adenine and cytosine, like any combination of two nucleotides repeats. That is AG, CT, CC, repeats. They are dinucleotide or trinucleotide, CAG, GAC, or GGA, or CCT, any of them. So these kind of repeats, these are the units. The repeats are, say, one human has at one place CCT repeated 6 times, whereas the other person has CCT, same CCT. He has, say, 14 repeats. Okay. So for example, myself at one place, I may have on one chromosome CCT 14, and then another chromosome I have CCT just 3. See? So if I have CCT 14 on one chromosome, CCT 3 on one chromosome, what it means? That my dad had 14, or my mom had 14, or my dad had 3, or my mom had 3. One allele came from dad, one allele came from mom. Okay. If I know my mother's genotype, I can infer which allele came from whom. Our father's genotype, mother's genotype will tell us how I received these two alleles. I'm heterozygous. So all of you are heterozygous for these repeats. So these dinucleotide or trinucleotide repeats They are there tandemly repeated short sequences. They are also called as simple sequence repeats. So there are plenty. They are everywhere in the genome. So SNPs we can make, we can sequence humans and align those sequences with the reference genome. And then we have one base pair, one SNP difference for every 1,000. Other slide says 1,000. This slide says 1,300, which is okay, about 1,000. So quite different we are. Though we are 99% same, these single nucleotide polymorphisms make us different. So there are chips made out of microarrays, chips of arrays. So these chips will contain 20 million infinite number of spots. Each spot is a length of sequence that differ for a SNP, using which we can scan human genomes. Using these chips, we can scan human genomes. Like I can take one drop of blood from you, one micromil of blood from you, and isolate DNA. And the DNA, I can use it to hybridize to this DNA microarray chips. These chips will tell us how your SNP configuration is there, what kind of SNPs you have in your genome. So it's like this. Say here is one individual, and he has on one chromosome, say mother's chromosome, TA, and on father's chromosome, TA. So we can say this is homozygous, okay? And this is another individual, exactly at the same position. This is John, this is Mary. Mary has CG at this position, whereas John has TA at this position. And CG is another position. And they produced a baby. Here is the child. So this chromosome came from dad, this chromosome came from mom. So TA is a SNP, and CG is another SNP, for which he is heterozygous. So this is how SNPs are very, very useful. Millions of SNPs, you are kind of noting this kind of signatures. Okay. So these SNP signatures or SNP genotypes are very powerful. So this person is TA, this person is CG, whereas this person is TACG, heterozygous. And then copy number variation is again intriguing. You know, sometimes regions, segments like 200 to 300 KB containing many genes are different. So many of the individuals are heterozygous for copy number variation. Some of them are deleterious, some have adaptive advantages. Do I have that picture? I don't have it here. Okay. So let me tell you one example, very nice example about copy number variation. It has adaptive advantages. We were all, some of our ancestors were hunter-gatherers. Some of us were going to hunting. Some of the tribes or some of the communities in the ancient human populations, they were hunter-gatherers. Some of them were scavengers. You know, before, during the Paleolithic era, people just collect food. That is fruits, vegetables, grains, they collect. They don't cultivate, they just go there and pick them up. There are some communities, they don't do that scavenging work or gathering work, food gathering work. They just go for hunting. Morning, They take their implements, stone cutters or sparrows, bows, stones, rocks. They just go after animals. They kill animals and bring home. So these communities diverged. Some are just gathering communities that whole family goes gathering fruits, nuts, vegetables, seeds, stuff like that, or leaves that can be edible. And some of the communities go for hunter-gathering. So what it has to do with the modern era, none of us neither go for gathering nor hunters. We just go and buy food. But those genetic signatures are embedded within our genomes. How can we say that? Some of us have this amylase, starch amylase, that is there in our saliva. That amylase degrades starch. Okay, so some of us have 6 or 8 copies of that gene. So if we eat a lot of sugar, say bunch of donuts, our weight will not increase because we have enough amylase to degrade that. So using that amylase, we can degrade all our starch that we consume. Okay. So that do not impact us. But the genome that is coming from hunter-gatherer communities, ancestors were hunter-gatherers, they have just 2 copies or 3 copies of amylase. They cannot effectively metabolize that starch. They are meant to eat more meat. But we eat the same food. A sandwich is made, some of it is a meat, some of it is starch. Our donut is full of starch. Our juice is full of starch. So in modern food, we have mix of food. So our communities don't differ that, hey, you eat only meat, I eat only starch. But the starch amylase, the genes that degrade those starch, the number of copies are clearly fixed in your genome. Like if you know that you are the person who cannot metabolize starch, you should not be eating them. If you eat that kind of starch, I mean, lots and lots of chocolate and sugar, you don't have starch amylase sufficiently to degrade all that starch. You see, that is one adaptive advantage. Or also some of the communities, our ancient communities, they use it to fight wars. All the time, 2 tribes fight. One community invade a village and kill all the women and children. Do lot of massacre and come back home and they wash their feet and legs and have relaxed sleep. Okay, and some of the communities that do not go for the war, they build fences around their village. They train their soldiers, or some of the guards will be trained, or they keep some kind of barbed fencing around their fields. They don't go for the wars. So what it has to do with the modern communities after several thousands of years, then the genome, our genomes are evolved differently, like number of copies that can withstand this kind of stress. Okay, so there are regions of the genome. If you have more copies, you can withstand a lot of violence. For example, some of our citizens, they go for the war. Say they go for the Iraq or Afghanistan, and once they come back, some of them will forget very easily. They just don't care. They just say, well, I went for a war and I am veteran. Forget about what happened in the war. But some of them will never forget whatever they have seen there. They get trauma, and that we call as post-traumatic stress disorder, PTSD. So why do they have PTSD? Why his friend or his colleague do not have it? Because copy number variation of the genes that make them to withstand that kind of violence. So these kind of copies are kind of selected upon for thousands of years and fixed in the genomes. So I have given two examples how using the number of copies you can identify what kind of human that could be, okay? Whether he could withstand the violence or not, whether that person can eat lot of starch or not. So that kind of studies can be done if you study this copy number variation. So once every one of our genomes, all of our genomes get sequenced, we can understand this profound knowledge, okay, about ourselves. We do not know ourselves. I cannot say from what kind of community I came from, whether my ancestors, they went to wars, or they were just gatherers, or they were hunter-gatherers, what kind of life they led. I don't have any idea until unless I sequence my genome and understand what kind of genomic signatures are embedded within me. Okay. So similarly, we discussed several times. I don't know in this class I discussed or not. That is called disruptive selection. There is a kind of selection. Why we need to hold Olympic Games separately for women or men? Why don't we have combined games? The reason is the genes within women are disruptively selected in the sense when the hunter-gatherers were there, much more ancient times, then men will go for hunting and they bring the meat home. When they kill an animal, they get to eat fresh meat. They can probably drink blood and eat some vitals and bring leftovers home. And even in the leftovers, every day breakfast, lunch, and dinner, they will get massive share because they are the ones they go for hunting. Whereas women, they are just to tend to cave or take care of the children. Their job is probably gathering nuts here and there and take care of the cave and children, or weave this dress for winter from the skins of the animals. So their job is kind of very simple job. So they don't get to eat lot of food. They were given some leftovers. So those genetic signatures are so embedded that it takes another thousands of years so that men and women are physically same, right? Several hundreds of years. So as long as we practice discrimination on anything, then inadvertently, we do not know how, without knowing, without our knowledge, we are imposing genetic selection. Whether you are imposing racial discrimination or any kind of discrimination you are doing massive genetic selection there. Okay, if you say, hey, this kind of people I would like to have, that means you are imposing a selection there. That is going to leave a genomic signature in your genomes. That takes massive amount of time to erase those signals. So in India, we have untouchables. Some of the communities, we cannot touch them, but not nowadays. Maybe 200 years back, you cannot touch some communities because they are ugly, untouchable people. They are meant for, field work, or they are meant for works that are not civilized people are supposed to do. This discrimination continued, say, 5,000 years, 6,000 years. Then what happened? You need to have different tests or different criteria for selection into schools and colleges because they are intellectually became inferior because of massive selection that was imposed upon. So for several hundreds of generations, they were asked, you are not meant for study. You are just meant for doing war or chiseling some rocks or building homes. And then the other communities, they are meant for education. They are scripts. They are writing, reading, or implementing laws. These communities intellectually enhanced themselves. So this kind of selection went on for several hundreds of generations. So this can happen anywhere. As long as you practiced slavery for hundreds of years, you need, but though that slavery time was very, very less, it takes relatively lesser time to recover those genomic signatures. But if you do that long time, thousands of years something, like thousands of years slavery, can impose a lot of genomic signatures that are imprinted in the genomes. So the copy number variation, single nucleotide polymorphism, these have profound knowledge we can gain about the human histories and the human capabilities. So this kind of polymorphisms are prevalent. There are lots and lots of repeats. For every 2 kb, 2000 nucleotides of the human genome, there is a repeat, and they have repeat length differences. So how to know what is the use of these polymorphisms? There are so many different differences in the humans, and what is the meaning of each and every difference is totally unknown yet. We don't know. At present, we have probably knowledge of only 10% of these differences. 90% of the differences we humans have, we don't have knowledge yet. We don't know what kind of phenotypic trait they are kind of associated to, until unless we do association genetics. So thousands of similar people need to be sequenced and need to be compared. Okay. For example, 1 million obese people can be recruited from all communities. And then 1 million people that are not obese need to be recruited several communities. And then massive sequencing has to be done and compared. And then associate the polymorphisms for the human weight. You see, that kind of studies have been done for the cancers and diabetes or weight, aggressive behavior. For many traits, you can do mapping these days using that genome-wide association mapping. So here is a repeat. This person has TGA 1, 2, 3, 4, 5, 6 times, whereas other person has 8 times. Other person has 10 times. And why this difference? How can you infer this difference? So you need to do a lot of association mapping to understand what is the utility of these differences, why we are having these differences. One side, we all look different, which is good, and that is because of these alleles. You know, these are all different alleles. Whatever polymorphisms we are seeing, they are different alleles. But what is the meaning of that alleles? We do not know until unless we study systematically that similar individuals share these alleles. So probably this allele may be useful or important. So humans accumulated a lot of deleterious alleles. We are baggage of a lot of deleterious alleles, bad alleles. And how to purge these bad alleles? You know, lot of new technologies are there, like genome editing or correcting the genome, stuff like that. So the human genetics is a highly evolving field. So especially the pedigrees can be analyzed, human pedigrees, and the SSRs and SNP differences can be noted. And also we can, if we don't have money for sequencing, we can do just digestion with restriction enzymes and see the digestion pattern, because digestion happens only at some selective sequences, known sequences. A particular enzyme can identify a selective sequence. So digesting with that enzyme, we can compare this fragment pattern, restriction fragment length polymorphism patterns. Okay, and that needs just a simple electrophoresis. So here is one important study, one simple study to understand the polymorphism. Here is mom. She has four repeats and 5 repeats. And here is the dad that has 4 and 6 repeats. So the baby has 4 coming from mom and 6 coming from dad, 4 and 6. Okay. So there is another family. Mom has just 1 repeat and on another chromosome 2 repeats, whereas dad has 3 and 4 repeats. So this boy has 1 repeat coming from mom and 3 repeats coming from dad. These two mated to produce all these children. Some are boys, some are girls. But now 1 allele comes from dad, 1 allele comes from mom. So 4 comes from mom, 1 comes from dad. 1 comes from dad, 6 comes from mom. 3 comes from dad and 4 comes from mom. 1 comes from dad, 6 comes from mom. 3 come from the mom and 3 comes from dad, 6 comes from mom. 1 comes from dad and 4 comes from mom. So you see, among these brothers and sisters, anyone is similar? All are different. All brothers and sisters, they are different. They are 7 people. All 7 are totally strikingly different. You see how nicely in gel you can resolve. This gel, whether it is 4 repeats or 6 repeats, 4 or 5 repeats, can be seen based on their migration. Okay. After doing electrophoresis, you can see it. So this guy has 1 repeat, 6 repeats. So much distance. And their children will be totally different as well. So this is how all these alleles get shuffled. If you are playing poker, one poker game is not exactly the same like another poker game. Can you say the same? No. A set of 10 games are played from one deck. That set will be differing from each play. It's like that, picking up 2 alleles and then matching them. Okay. So you guys can take 10 minutes break, and during the break, I will interact how the lecture has been. Okay.
What do you think of it about this? Things. I really suck at things. There's some years on there. I guarantee I never every single one of them. Like who did what, and I'm like, if we're talking, I can tell you. But if I'm reading it, I will gaslight myself every single time and tell myself that is the
Okay.
Yeah, that's wrong. It can't be that. That's too easy. Yeah.
So, okay, folks, back to our Neurospora crassa. We understood Neurospora crassa. Let me see if I put it in pause. No, it's good. Good to go. So the Neurospora crassa is an ascomycetes. It's a haploid. Okay. So it produces conidia, conidia spores. Conidiums are asexual bodies, and they have cells inside. Those cells will mature and become conidia. But they are also capable of having sex. There are females and males, mating types. So females will have archegonia, which will have an egg, and the males will have antheridia, which will have sperm. So the nuclei can swim through and find the egg and fertilize to form a zygote. Okay. So this also can be used for genetic mapping. So working with ascomycetes, just like Saccharomyces cerevisiae, Neurospora, you can map these genes profoundly because life cycles are very, very short. And then you can grow large numbers of families, progenies. Okay. So one thing we need to understand here, what is a tetrad? Okay. So meiotic products, once the meiosis is over, the zygote especially is a 2N. and 2N zygote undergoes meiosis, and this meiosis will have 4 different cells in the first meiosis. They are called unordered tetrads. We'll discuss why we call it as unordered tetrad. Ordered tetrads are in the second generation. In the second generation, all these 4 becomes 8 tetrads, and these 8 tetrads are placed in such a way that we can understand the geometry of meiosis. That also we will understand. This is very important word, geometry of meiosis. We cannot understand geometry of meiosis in other organisms like animals or plants, but we can understand in fungi. The reason is fungi is life cycle is small. You can grow them in large numbers. So one thing is 3 different kinds. Parental ditypes are just looking like parents. Or non-parental ditypes are not looking like parents, but there is no recombination involved. Okay, but all of them are looking like recombinant combinations, but there is no crossover there. Tetratype are half of them have undergone crossover because of chiasma formation. We will discuss a little bit more detail and come back here. There are 2 homologous chromosomes, mother's chromosome, father's chromosome. They have not undergone and they just separated. Okay, but here crossover happened, and then you have a recombinant. Let me see. Where is that other figure? Oh, unfortunately, other figure is not there. Yeah, here it is. Okay, here is 2 homologous chromosomes. And these 2 homologous chromosomes are going to segregate in meiosis. One homologous chromosome goes to one place and other goes to one place. And when the spore formation happens, you have ascospores with different chromatids. This is called ascus. Okay. Here we should have put another slide to understand 2 different genes. Let me see, annotate. Here itself I can try. So for example, this is another chromosome. It has B on it, capital B. And then small b. Okay. So when they form chromosomes, you have 2 chromatids. This has capital B, 2 chromatids. Full chromosomes are formed from the chromatids. And this one has small b, small b. Okay. Now the segregation can happen in such a way that this capital A and this capital B can form a spore, say capital A containing chromatid, and there is another one, capital B. Okay. So capital A and capital B are together. And this can also form capital A, capital B. One is capital A, another is capital. Whereas these things will form small a, small b. So these are exactly like parents. One parent had capital A, capital B, another parent had capital A, small a, capital A, capital B, another parent has small a, small b. So these are exactly like parents. Two are like this parent, two are like this parents. These are called parental ditypes. Since they are looking exactly like parents. Here, carefully watch, folks. Now this is small b is here. Because either that's the equal chance for capital B chromosome to be up or small b chromosome to be up. So here is this chromosomal arrangement is such. Here is capital B. Small capital B is down here. In previous one, this chromosome was up here. So this chromatid and this chromatid can come together to form capital A, capital B spore. And then another spore was other parental kind, small a and small b spore. But now this arrangement, what arrangement? This capital B chromosome came down and small b chromosome came up in the meiosis equatorial plate, metaphase equatorial plate. Now the division will be capital A is combined with small b. Maybe this is better to write it fast. Capital A, small b. Or small a, capital B. Small a, capital. So these things can form. What happened? All of them are recombinants. Whether there is a crossover happened, there is no crossover. Only the chromosomal arrangement. Okay. So the actually capital A, capital B, the original parents, let me draw original parents here. This is one parent. This is one parent. Okay. So these two are on. These chromatids doubled. So that is one. Because they are haploid, right? The fungus, they are haploid. So it became like AB. And then AB. So on the equatorial plate, this capital A this chromosome and this chromosome will be capital A, capital B will get selected. But what if they are arranged in different fashion, like this? So one way is whether they will get arranged. You know, chromosome with the capital B's are up there. So this can make all parental ditypes because this chromatid can join together in one spore, making capital A, capital B, capital A, capital B, small a, small b, small a, small b. But if it is just opposite way arranged, if this is not arranged this way, on the equatorial plate, if the arrangement is different, how different? That is, small b is here, not capital B.
All right.
And then here also, small b is there. Okay. And this is capital B. Sorry. Capital B are here, small b are up there. This is going to create like this. All non-parental ditypes. If you are seeing 100% non-parental ditypes or 100% parental ditype, what are you going to conclude? I am asking you. Or let me ask another type. Parental ditype is exactly equal to non-parental ditype. What it means? What do you infer from that? Both genes are on different chromosomes. That is A and B are in different chromosomes. So this, why did we get all 100% recombinants here? Because they are all non-parental ditypes. They are not like parents. Because the way they got arranged on the equatorial plate, either capital B can be up there, small b can be up there. That's random. No one can decide. So 50% of meiosis will end up parental ditypes. 50% of ascospores will end up non-parental ditype, telling us that both these genes are not linked, but are on different chromosomes. But if they are on the same chromosome, so what can happen if they are on the same chromosome? Then the question of linkage comes. So say, for example, B is here, capital B is there, and then small b is there. Then comes the question of recombination that can happen here. That is B. This is small b. Here is again one more capital B. Small b. So the recombination is happening between the internal chromatids. One is having capital B, capital A. One is having small b, small a. Recombination is happening. And then two parental types are there. This is capital A, capital A, capital B, and this is small a, small b. But this is not like that. This has capital B, capital A, capital B, a, small b, but has undergone recombination. So as a result of that recombination here, this is parental ditype. This is another parental ditype, whereas this is recombinant. And this is recombinant. Folks, can you explain it? Why did it happen? Because initially the one parent had capital B small a because they are haploids. One chromosome is enough. Other chromosome coming from other parent had small b small a. You mated them. In zygote they came together. Each chromatid made full chromosome. Okay. So one chromosome has capital A, capital B. This chromosome also capital A, capital B. This one had small b, small b, small a, small b, small a, small b. Let me go. This chromosome, capital A, capital B. And this side, small a, small b. This has small a, small b. Indicating what, folks? There is no recombination here, but the recombination can happen. This capital, hopefully, oops, alga. Okay. The recombination can produce a chromatid that is capital A, small p, or small a, capital B. You agree? The recombination can happen between these two. Okay. And then segregation can happen as a result. You have that chromosome with capital A, capital B, small a, small b, small a, capital, these two are recombinants. So you have parental types as well as recombinants. So this kind of ascus is the whole thing. This is ascospores. It's called tetrads. So pertaining to two different genes, in ascomycetes or neurospora crassa, you made a cross. If all of them are non-parental types, all the ascus, and 50% of them are parental ditypes, 50% of them are non-parental ditypes, you are going to say they are on two different chromosomes and assortment is just like independent assortment. But if they are on the same chromosome, that means you will have both parental types as well as non-parental types. Ascospores, both in same ascus. The whole thing is called ascus. So here, if both chromosomes are, one is parental type, and this is non-parental type, then we call it as tetrad. If tetrad is there, that means they are linked. They are on the same chromosome. Now, what is the linkage? How do we estimate linkage? Number of recombinants, total progeny. Here, number of tetrads to the total progeny. Yeah. So we understood parental ditypes, non-parental ditypes. What are the parental ditypes? 100% parental kind and 100% recombinants. That can only happen when the two genes are on different chromosomes. Whereas tetratype or tetrads, you can call tetrads or tetratypes. You can observe all four genotypes. That is parent one kind, parent two kind, and then recombinants. So the existence of TT, that means there is linkage. They are located on the same chromosome. The number of TT divided by total number of tetrads. Sorry, tetrad is, any ascus is a tetrad. Sorry for that. Tetratype in the sense having both. Two spores are parental kind, two spores are non-parental kind. They are called tetratype. So tetratypes means TT. Number of TT divided by total number of ascus times 100 by half. That gives linkage, mapping distance. Because fungus, like eukaryotes, they don't have a lot of junk DNA. So mostly genes are collinear. So the genetic mapping and the physical mapping corroborates really well, unlike eukaryotes. So the ordered tetrads, PD, NPD, TT, these are all the ordered tetrad in the sense in meiosis II, all these tetrads become two, right? They kind of, I will show you the figure, yeah. So here is the first stage, recombination happened, and now you have meiosis II. So in meiosis II, one possibility is all capital A's and small a's are next to each other. Okay, with reference to single locus. Arushi, this is very important, not it. There is second division segregation that gives geometry of meiosis. Geometry of meiosis, we are not referring to two loci within the single locus. What are we trying to understand in the geometry of segregation is how far is the gene from the centromere. Okay. This kind of centromere mapping is not there in plants or animals. So if it is very close to centromere, then the segregation will be like this. All one parental kind ascospores, and other parental kind spores, either this way or this way. But if there is recombination, swap between the chromatids, that tells they are further away from the centromere. This statement is very critical. Once they are further away from the centromeres, the chromatids can undergo recombination. And you will find various arrangements here in the ascospores that have various different arrangements. You see, all four came here flanking with two dominant alleles here, are shuffled, or in middle there are dominant alleles flanked by recessive alleles. So now you can count ascospores that are having this kind of arrangement and this kind of arrangement. This gives number of asci. In the sense, ascus is in plural, it is asci with second division segregation to the total number of asci times 100. Okay, this is much more precise mapping. This gives how the meiosis II is arranged between the centromere and the gene. Okay, so that is very profound. And then I wanted to show you a video, if I can show you in the YouTube.
confidence boost when filing your own taxes? Add a tax pro review when you file online with H&R Block. A tax pro will ensure you the best possible outcome and then file your return for you. Tax pro review. It's the convenience of home and the confidence of a pro.
So what I'm trying to show is, these are two DNA strands of one chromatid. This is other two DNA strands of other chromatid. Okay. So the crossover happens at the time. What happens during the crossover? There is a protein. There is a protein called the MRX that binds here. So once this on one strand, and especially on one chromatid, the DNA is resected by a protein. And then these are two other proteins, they cause strand invasion. So this is called D-loop. So what happened in the D-loop? That 4 DNA strands of 2 chromatids came together and resection occurred. And then invasion happened. That is, the DNA strands of opposite chromatid got fused up. So this DNA belongs to other chromatid that fell upon DNA strand of another chromatid. Now the synthesis begins. When synthesis is happening, here this segment is synthesizing using this as template. This DNA is getting synthesized using this as segment. So this time what happens? We imagine that there is a disagreement. There is a SNP located here. Say this has A here. And then this was T here, which was good. When both were together, both were homologous. Whereas this chromatid is of different allele. And this had G here, and this has C here. See? Now it is AG here, TC here. But they are not complementary. Only A and T are complementary. But A and T were DNA of one chromatid. But during the crossover, now this is swapped. At this point, what can happen? Whether this G is removed and replaced with T, or this A is removed and replaced with C, so that there is a complementarity is restored. And similarly, this T is removed and replaced by G, or the C is replaced and removed with G. So this junction is called D-loop junction, is named as Holiday junction. Why Holiday? Because Robin Holiday is the scientist who identified this D-loop. And what forms are the D-loop? Why D-loop has to form? Because when two chromatids are coming together, each chromatid has 2 DNA strands. When 4 DNA strands entangle and make a loop-like structure, and here the resolution should happen. That's why this is called Holiday junction. This junction is crossover junction is called Holiday junction. You can happen this way or that way. Now the DNA level exchanges happen. So folks, what happens? Gene conversion can happen. You know, when 4 A's and 4 small a's are there in the 8-spore cell, some will get swapped because of the gene conversion that I have shown you. Sometimes you will see 3A and 1A, or 1A, 3A, because the alleles are converted into one or other fashion. When A and G are combined in the Holiday junction, either A is removed or G is removed. So the alleles are being shifted to capital A allele or small a allele. It's not segregating exactly the same way. Sometimes you will see 5A, 3A, 3A, 5A. All this kind of aberrant ones you can see in the gene conversion. In other words, what happens at the Holiday junction, when they A and C are combined, these are called heteroduplexes. You know, these kinks should not form because they are not complementary. But crossover junction created a scenario where the alleles of opposite chromatid came together at the molecular level, and they are different. So either A is swapped to G or C is swapped to T. So these conversions do form. So this is cause for gene conversion. And then we understood Holiday junction, why the T loop forms. Because 4 strands will be there together at the chiasma. All these 4 strands can be resolved by 2 different proteins to form D loop. So the D loop, once see the blue is coming from one chromatid, and then red is coming from another chromatid, red DNA. And then both got fused like this, 1 strand of red and 1 strand of blue, at which a lot of gene conversion will happen. And after resolving, resolution can happen in different ways. There is enzyme resolvase that can resolve like this. Okay. And then you have this blue strand sticking to this red strand, or like this. That means part of blue strand, part of hybrid strands, part of blue, part of red, part of hybrid, part of hybrid, red. So you have all kinds of combinations here. Site of crossover, how it gets resolved, how the gene conversion can change. All this matters. Okay. So this is really a chapter that you guys need to think about, read many times, and discuss with me several if you need any further clarification. So there has to be mutual effort to understand what is happening at the crossover junction. Okay, this is really a very intriguing topic to understand how the crossover is resolved at molecular level. Thanks, folks. Good night. Class is resolved.